We're moving away from the general molecular biology material, but we still have a couple of more basic overview general lectures to do before we start talking about all of the various molecular techniques. So today we're going to discuss genetics. At the end of this lecture, you should be able to list the main characteristics of the human genome and define numerous genetic terms that are listed on this slide. This slide shows a diagram of the cell. And remember, we have our human cell, and inside a eukaryotic cell, you have your nucleus, and inside the nucleus is all of the genetic information. That's where the genome is located. And within that, you have chromosomes, and that's going to be the bulk of this lecture. So remember, we have our biological information, which includes DNA, RNA, and protein. DNA is a blueprint of all living organisms, and in the eukaryotic cell, it exists in the nucleus, and it's condensed into chromosomal structures. For our RNA, we have various types of RNA: transfer RNA, ribosomal RNA, and messenger RNA. And messenger RNA is the material that is translated into protein. Proteins are the workhorse of the cell. They're what does all of the various functions that are essential to keep that cell alive. Gregor Mendel is credited for being the discoverer of modern genetics. Genetics is the study of how information is transferred, how traits are expressed, when traits are expressed, and the various mutations and abnormalities that can occur. So when we talk about genetics, we have to talk about genes, and genes are the units of genetic information. Each gene provides the instruction for a particular property or characteristic. In lower organisms, such as your bacteria, there may only be 2,000 to 4,000 genes, whereas in higher organisms, there are many, many more. Now, chromosomes, your genes are within the chromosome, and the chromosome are within the nucleus of the cell. Lower organisms fit all of their genes on one chromosome. However, higher organisms have so many more genes that they need several chromosomes. Chromosomes are distinguished from one another in the cell by the size, the location of the centromere, and the unique banding patterns. And usually you're going to look at the banding patterns using special staining techniques. So this slide shows you the morphology of your typical chromosome, where you have your telomeres at the end, the centromere in between the telomeres, and you have two different arms. You have the short arm, which is called P, and the long arm, which is called Q. And the chromosomes can vary based on where that centromere is located. So if it's directly in the middle, it's metacentric. If it's a little closer to one end, it's submetacentric, and if it's almost at the very tip, it's acrocentric. So we already said you can look at chromosomes in more detail by looking at the banding patterns, looking at the size of it, looking at where that centromere is located. Chromosomes can be visualized using various staining techniques. So the Gametes staining produces G bands, and that's the Gametes stain stains heterochromatin. Now, harsher treatment of the chromosomes before Gametes staining will produce R banding, and that stains the euchromatin. Alkali treatment of chromosomes results in C banding or staining of the centromere. Now, the problem with some diseases is that there are abnormalities in the chromosome structure. So we have translocations, which is where you get a portion of one chromosome that's going to go into another chromosome. You have deletion, where part of the chromosome just disappears. You have inversion, where one part of the chromosome moves to the other side of the chromosome. You have isochromosome, where the chromosome can split. You have derivative chromosomes, where you can get various shapes and different pieces of other chromosomes that attach. You could have insertions, where you insert material that shouldn't be there, and there's ring chromosomes. So the human genome is comprised of 46 chromosomes. There are 22 autosomal pairs, which make up the 44 chromosomes, plus your sex chromosomes, which is your X chromosome and your Y chromosome. There's a total of 3 billion base pairs in the human genome. There's approximately 20 to 25,000 genes. Now, with 3 billion base pairs in human DNA, it was thought that there'd be a lot more genes than that. But in fact, only 1.5% of the total genome serves as a protein coding region. A karyotype is a complete set of chromosomes in a cell. So in the human cell, we already said we have 46 chromosomes, 44 autosomes, and these are chromosomes 1 through 22, and then the 2 sex chromosomes, which is your X and your Y. This is a schematic of the human karyotype, where you see your 22 autosomes plus either you get an XX or an XY to make your 46. Here's another picture of a karyotype. Now keep in mind we have our genome, which consists of your 46 chromosomes. Each individual chromosome has genes, and these genes are separated into, or your DNA is separated into your exons, which are your coding regions or your genes, and in between the exons you have intergenic DNA base pairs, which is called your intergenic region or your introns. And remember, in eukaryotic cells, when you transcribe your DNA into your RNA, that intergenic region or the introns are spliced out so that you just have your coding sequence or your genes or your exons one after the other, because you don't need that intergenic region when you are making proteins. So genes are expressed, and when you're referring to someone's genotype, that is their actual genetic makeup, what those genes are. The phenotype is what we see, or the observed trait. So the phenotype is based on that genotype and how those genes are expressed. Each pair of genes forms an allele, and each one might be dominant, recessive, or a combination. So your alleles, each gene exists in alternate forms. So different alleles of the same gene are related, but they may have minor variations that can produce different outcomes for the same characteristic. So our traits, not all of our traits are due to a single allele. Some are the result of a combination of several alleles. And this accounts for the ranges in expressed traits. So depending on how many of those alleles are being expressed and their amounts of expression, you get variation in hair color and height. So two different people may have the genotype for brown hair, but one person has a lighter brown and the other person has a very dark brown. And it's due to this combination of several alleles and their expression levels. So, of course, we have our definitions. Haploid is having only a single copy of each gene, and this is common in lower organisms. Diploid is having duplicate copies of each gene. When we refer to something as being homozygous, these are having two identical alleles, and heterozygous is two different alleles. So remember, our somatic cells in eukaryotes are diploid. However, your gametes or your germline cells, your sperm and your egg, these are haploid. And this occurs through meiosis. So meiosis, you make haploid gametes from the diploid parental cells. So it's a matter of chance as to which original pair of copies gets passed on, which is why siblings vary in their phenotype, in the way that they look, the way their genotype is deciphered. Your zygote is that cell that's formed when the sperm and egg unite, and that's what forms a new individual. So remember, in meiosis, you have two different stages of cellular division. So in the first stage of cellular division, that's just like mitosis, where you get two diploid cells. From those two diploid cells, that's what you would get with a somatic cell. You then get another stage of division, and that forms four haploid cells. So mutant versus wild type, and you see that a lot of times when you're reading papers. So wild type is the properly functioning gene. So it's the original version, which is from the wild, which is called wild type. However, if that gene mutates, then it's considered different than the wild type. So you get a mutation or a genetic alteration. A null allele is a mutant allele that results in the complete absence of protein. And proteins very often are enzymes. So many of our inherited diseases follow Mendelian genetics, although not all of them do. In Mendelian genetics, you have dominant alleles and recessive alleles. The dominant allele is expressed at the expense of an alternate allele. So the phenotype that's expressed in the first generation is from that cross of two pure lines. And a recessive allele, the expression of that allele is going to be suppressed if there's a dominant allele present. The phenotype that disappears in that first generation from the cross of two pure lines is the recessive allele, but it can reappear in the second or F2 generation. So we have a genetic cross here of big D, and little d, little d. So you get one from one parent and one from another. So let's say in this case you would have to get one big D and one little d. So that first generation, or the F1 genotype, is going to be big D, little d. There's no other possibility of the genotype in that first generation. So we have our ratios. So if you do a typical Punnett square where you have your big R, little r crossed with big R, little r, you're going to get, in the gray shaded regions, big R, big R, big R, little r, big R, little r, and little r, little r. So if this were the genotype for the red phenotype, 3/4 of have a possibility of being red because three of them have the big R or red genotype. 1/4 is going to be white because that's little r, little r. The sex determination of an individual. So this is determined by sex chromosomes. So in a female, you have 2 X chromosomes. In a male, you have 1 X chromosome and 1 Y chromosomes. So your egg cells carry a single X chromosome. Your sperm cells, half of them have a single X and the other half have a single Y. So it's going to be the sperm cells that are going to determine the sex of an individual. So here's a Punnett square for sex determination, where you have big X, big X, big X, big Y. So you have an XX and an XY that crosses. 50% are going to be males and 50% are going to be females. A gene is considered sex-linked when it's carried on one of the sex chromosomes. And there are some hereditary diseases that are called sex-linked diseases. So here's a schematic of an X chromosome, and you could see there are many different regions on this X chromosome, and these different regions have various genes, and these genes are going to be the genotype for many different characteristics. Here's your Y chromosome, again, various regions that have genes in them that perform specific functions. You can have variations in dominance. So we talked about dominant allele and recessive allele. Well, you can have what's called partial dominance. And this is when a functional allele partly masks a defective allele. So that defective allele can come into play. In codominance, this is when you have 2 functional alleles that both contribute to the observed properties. Linkage occurs when 2 genes are on the same chromosome and they can be linked together. Crossing over results in recombination, which we discussed previously. So this is when 2 different strands of DNA are broken and then joined to one another. So what you get is the shuffling of various alleles between the chromosome. The farther apart 2 genes are on a chromosome, the more likely a crossover is going to occur. So here is a schematic of crossover where you have your chromosomes and one region of one chromosome is going to cross over and swap with the other region of the other chromosome. And so you get a shuffling of the genes. So during replication, that's when crossover occurs. And crossover is very important because it assures that the offspring differs from the parents. So in summary, mutations are heritable changes in DNA, and they can include changes in the chromosome number, structure, and gene mutations, which we will discuss in the next lecture. Chromosomes are analyzed through special staining, such as Gametes staining and karyotyping. Karyotyping will detect changes in the chromosome number and structure. And structural changes of a chromosome include translocation, duplication, and deletion.